Hello, BISC 10 students. Today we're going to cover Chapter 2, Basic Biochemistry. It's not too overwhelming, but there are some really important basic concepts that need to be looked at. So let's get started. So what are molecules? Molecules are formed when you have two or more atoms that bond together. An atom is an example would be hydrogen or oxygen, anything on the periodic table, essentially. But when you put them together, then they create something called a molecule. So individually, they're considered elements, and then put them together, that's a molecule. Now, a molecule, a really simple one that everyone pretty much knows, is H2O, water. 2 hydrogen, 1 oxygen. That's considered a molecule because it has 2 or more atoms bonded together. Now, compounds are molecules that are made of more than one type of element. So you could say that glucose, C6H12O6, is a molecule, but you can consider it a compound because it's made up of more than one type of element. I don't expect you to know too many examples, but knowing a molecule is something where you have multiple atoms forming together, or bonding together, to form a molecule is pretty important. We're going to look at organic molecules in this chapter. Organic molecules are molecules that specifically have both carbon and hydrogen in their structure, in their chemical formula. This example right here, C6H12O6, is glucose. Okay, it's glucose. There's a whole bunch of others. Methane, which is CH4, carbon, 1 carbon, 4 hydrogen. There's a lot of other sugars like sucrose and fructose that have the same kind of CHO formula as glucose. The numbers may be different, but they have a similar structure. They have a similar arrangement in terms of atoms: carbon, hydrogen, and oxygen. We're going to look at the 4 major organic molecules one by one. Now, before we get to that, there's some other things that we need to talk about first, but we will get to organic molecules, and it's very important that we discuss them. I want to talk about a couple concepts called the terms hydrophilic and hydrophobic, as well as amphipathic. Hydrophilic, hydro means water, philic means loving, so if you break the terms down using Latin, it's hydro is water, philic is loving. These are substances that will dissolve in water. Any type of polar molecule, like water, will break down in water, like salt or sugar. Okay, they have polarity, so they can break down. Sodium, NaCl, has ionic bonds, and when you drop it into water, it dissolves, right? If you've ever put salt in water, it dissolves. That's because of the charges associated with water on the water molecules, as well as the charges associated with the sodium molecule. When you drop those in, they dissolve readily. Hydrophobic, on the other hand, is water-fearing. They do not readily dissolve in water, and that's because they are non-polar or non-ionic, meaning they don't have a charge. A good example of that would be a stick of butter. Butter is made up of fatty acid tails and glycerol, and when you put it in water, it doesn't dissolve because there are no charges associated with that structure. Therefore, water can't separate the tails or any of the other pieces of the structure of butter, for instance. You could also say the same thing for vegetable oil or olive oil, peanut oil, etc. So hydrophobic simply means water-fearing. It's more along the lines of just it doesn't dissolve within water, whatever that substance is. Now, amphipathic molecules are molecules that have both characteristics of hydrophilic and hydrophobic substances. We're going to talk about a molecule specifically associated with cell membranes that has amphipathic traits. It has both hydrophilic and hydrophobic regions. Whoops. There we go. Here we go. So this is a perfect example. Having both hydrophobic and hydrophilic regions or properties. The head of a phospholipid is hydrophilic, while the tail is hydrophobic. Now, this is why when you pour oil into water, it forms bubbles. Because what happens is the heads of the specific grease or oil will face outward while the tails face inward, creating a bubble around the grease. Now the key here is that the tails cannot come in contact with water, which is why they create a bubble. The grease is right here. What we call a myocil, it's just basically a bubble around the grease, forms where the heads are facing outward and the tails are facing inward. Now, in cells, whether that's bacterial cells, plant cells, animal cells, doesn't really matter. The bilayer, the phospholipid bilayer that we have that makes up the cell membrane has, in the name, bilayer, two layers. So there's water outside a cell and there's water inside a cell, right? And if the tails can't come in contact with water, they repel water, then cells have to come up with a way to create a membrane that will be able to interact with both inside and outside the cell, the water in and out. And they do that by creating this bilayer right here. This outer layer faces outside, only the heads face outside, and the tails face inward. Now, inside the cell, where there's also water, another layer where the heads are facing inward and the tails are facing outward will form. So you get this what we call a phospholipid bilayer. This is what makes up a cell membrane, which is why we call it an amphipathic molecule, because it has both hydrophobic and hydrophilic properties. Now we'll talk more about bilayers in a bit. So chemical reactions. So chemical reactions are essentially breaking stuff and building stuff, breaking stuff and building stuff, and specifically breaking chemical bonds. Now when you look at a chemical reaction, you have the reactants on the left side, which are the starting materials, and the products on the right side. For instance, cellular respiration, where we make ATP, energy, we have glucose and oxygen on the left side. Those are our starting materials. And then on the right side, we have ATP, H2O, and some and ADP CO2 molecule. So you have your ingredients on the left, products on the right. They're what you create. The products are what you create. Now you could look at any chemical reaction and this would apply. There are some chemical reactions which are reversible, meaning they work in both directions, which actually the cellular respiration does work in both directions in terms of making and breaking ATP. So what about the rate of reactions, right? Rates of reactions fluctuate. Sometimes cells need more of something, sometimes they need less. And a lot of people have this misconception that reactions just simply go at a given rate at all times. And that's not true. Think of rates of reactions more along the lines of a dimmer switch, right? They could turn it and speed it up, turn it, speed it, or slow it down. Okay, they could slow it down. It all depends on the need of the organism or the cell. If a cell needs a specific molecule or biochemical biochemical molecule, then they can speed up the production of it. Or if they have too much of it, they can slow it back down instead of wasting energy. Which is why it's important to understand that rates of reactions fluctuate. Now, a catalyst is something that will help speed up a reaction. So let's say a cell needs to make lots and lots of glucose in a short amount of time. It needs to make it very quickly. What they can do is they can add a catalyst to the reaction, specifically an enzyme, and it will increase the rate of the reaction by speeding it up. Now there are some specific mechanisms that will allow this to happen. We're not going to go into too much of the mechanistic point of view of this. We'll talk about enzymes a little bit, but I don't go into a ton of detail. That's more biochemistry than this course. But it's important to know catalysts speed up reactions. Now, there are two major reactions that I want to talk about. First is dehydration synthesis, which is what we call a synthesis reaction or an anabolic reaction. And what that's referring to is the fact that you're utilizing energy to build something. So anabolic reactions use energy to build something. And specifically, dehydration synthesis is the reaction. So, if we take dehydration synthesis and think about it for a second, dehydration, what does that actually mean? Well, when you're dehydrated, what are you losing? You're losing water, right? And then synthesis means to build or create. So, dehydration synthesis, you are losing a water molecule or multiple water molecules to create something. And that's what this image shows right here. This is an amino acid chain right here, and here's an unlinked amino acid, and we want to join them via a chemical bond. Well, we use a dehydration synthesis reaction to create a bond between the endmost amino acid and the unlinked amino acid, and then you end up with a bigger one. And we keep doing that to build proteins. We add an amino acid, then we'll undergo dehydration synthesis again and add another amino acid, and another, and another, and another, so on and so forth. So it's important to know that anabolic means using energy to build something, and we do that via dehydration synthesis. Now, for every reaction, there has to be an opposite reaction. So for something to be built, we also have to have a way to break it down. And this is the same for humans and bacteria. So the opposite of dehydration synthesis is hydrolysis, which is a catabolic reaction or a decomposition reaction. Hydrolysis, if we break it down, hydro means water, lysis means break. So you use a water molecule to break the chemical bond. Now, from biology, if you've ever had it before, general biology, or even chemistry, chemical bonds store potential energy. Energy that can be used to power reactions. So, when you have a hydrolysis reaction or a catabolic reaction, you break a chemical bond using a water molecule, and the energy that was in that bond is now released to be utilized for whatever function the cell needs it for. Right? That's something that you really need to keep in mind, is that with hydrolysis or catabolic reactions, energy is released because we are breaking the chemical bonds, and that energy then can be harnessed to carry out some function. Right? And a lot of reactions are cyclical, meaning you build it and break it, build it and break it over and over and over and over again. And the two reactions you use are dehydration synthesis and hydrolysis. All right. Now there's a concept called activation energy that we need to talk about as well. Activation energy is essentially the amount of energy it takes to start a reaction. Activation energy is the amount of energy it takes to start a reaction. Think of it this way. You have a bundle of sticks, you're camping, and you're trying to start a fire. Right? And all you have matches and some gasoline, just a little bit. So you try to start the fire without using the gasoline, right? But it takes a ton of energy to actually start that fire. So the matches are not necessarily enough. So the next thing you do is you toss a little gasoline on it and light the next match, and the fire starts. Basically, the matches alone couldn't provide enough energy to start the fire or the reaction. Same thing here, same concept here. Every reaction has a given amount of energy needed to start it. So it costs energy to start the reaction. Now, the activation energy can be manipulated, meaning we could decrease the amount, which would speed up the reaction. And to do that, we would use an enzyme. Okay, we would use an enzyme. And we will come back to this a little later. All right, so recap quick. Dehydration synthesis, right? You're building something, right? You're building a larger something. Hydrolysis, you're breaking the chemical bonds, releasing energy. And then activation energy is the minimum amount of energy you need to start a reaction. All right, so let's go back to the biomolecules or the organic molecules. There are 4 of them that we're going to talk about, 4 groups. And it is very important that you know what we call the monomer of each of them. The monomer is the most basic piece of a specific biomolecule or organic molecule. And I'll give you the exact examples in a minute. But you need to know what the monomer is versus the polymer, meaning what the individual pieces are that are put together to build the bigger something. The 4 major classes are carbohydrates, which are sugars, nucleic acids, which is DNA and RNA, proteins, which has so many different functions. Proteins are incredible. And then lipids, which are fats. Now, when it comes to carbohydrates, we use as the building blocks, the monomers are simple sugars, meaning monosaccharides or disaccharides. Monosaccharide means 1 sugar. Disaccharide means 2 sugars. So the building blocks are monosaccharides or disaccharides. It's a simple sugar. Nucleic acids, the building blocks are nucleotides, and there are 4 of them in DNA and 4 of them in RNA. Now, there is a difference between DNA and RNA. They share 3, and then RNA and DNA each have 1 that's unique to each biomolecule. So DNA has 4, but there's one of them that is not found in RNA. RNA also has 4, but one of them is not found in DNA. So it's vice versa. Proteins, the monomers are amino acids. And then lipids don't necessarily have an official monomer. We could technically say the building blocks are fats and oils, which is correct, but they don't have monomers in the true sense of individual pieces that build together to build a larger something. So if I were to ask you what's the monomer of a lipid, technically nothing. There isn't one. If I were to ask you carbohydrates, you could say a simple sugar or a monosaccharide. If I asked you nucleic acids, which are DNA and RNA, the building blocks are nucleotides. They're nucleotides. And then if I asked you proteins, the building blocks are amino acids. So we'll go through all of these and talk a little bit about each of them. So the first one, carbohydrates. I mentioned that carbohydrates are sugars. The carbohydrates have this formula, CHO, and it looks like this: CH2O, and then with an N on the outside. The way that's written is fine, but I like to write it a different way. I prefer to write it as C subscript N, H 2N subscript, O subscript N. N is the number of those particular atoms. So for glucose, C6H12O6, it would have been, so C6H2 times 6 gives you 12, and then O, since it has N, would have been 6 as well. Essentially what I'm doing, basic math, distributing the N to each element. So N gets distributed as a subscript to carbon, as a subscript to the hydrogen, and then a subscript to the oxygen. And that just tells you the number of, if you have the number of carbons, then you know the answer is the same number of oxygens and double the amount of hydrogens. This isn't always the case. There are exceptions. But if you look at most sugars, they're going to follow that structure, or that formula, excuse me. Now, carbohydrates are really important for lots of different functions, mostly as energy sources, but they also are useful in structural components. You find carbohydrates in the cell membrane, or if you're looking at a bacterial cell that has a capsule, for instance, or a slime layer, what we call a glycocalyx. That's made of sugars that surround the bacteria. We'll talk about that in a minute. But here you can see we have two monosaccharides, right? Individual pieces. We want to put them together to create a disaccharide. And this shows us that if we want to put these two things together, we use dehydration synthesis to get this as our product. The disaccharide and some water. Then the opposite can happen. You can take a water, break this bond between the two sugars via using hydrolysis, and you go back to the individual monomers. So like I said, it's building them and then breaking them back down. Building them and breaking them back down. Over and over and over and over again. It's constantly happening. In bacteria, in our cells, it is constantly happening. These are some functions of carbohydrates. This is not comprehensive, but it is useful to know that we use carbohydrates mainly as an energy source, Structural biomolecules, meaning our bacterial glycocalyx, which is a outer covering around the bacteria, which we'll talk about in a minute. They can also assist in cellular adhesion. Cells can stick together because carbohydrates tend to be sticky. Communication. We can use them as communication molecules. And then there's also something called quorum sensing or environmental sensing that bacteria that live in biofilms can do. We're not going to really worry about that one. But these are just some examples. It is not a comprehensive list. Here's some specific examples of carbohydrates. Cellulose is found in plant cell walls. It's what makes up the cell wall of plants. Excuse me, of plants. Chitin is another structural polysaccharide. Basically, it makes up the cell walls for fungi. Also, chitin is really interesting because crabs, crayfish, lobster, crustaceans, essentially, their outer exoskeleton is made up of chitin. So that hard outer shell that they have around their, their muscle tissue underneath is made up of a sugar. Now, it's not exactly chitin like fungal cell walls. It's a derivative of chitin, but nonetheless, it is kind of a sugar-based structure that hardens. Very interesting. Peptidoglycan. Peptidoglycan is one of the most important structural components of a bacterial cell wall. Without peptidoglycan, you have little to no chance of having a cell wall. You may still have a cell membrane, but without peptidoglycan, there is no cell wall. If something ends in ose, O-S-E, it's almost always a sugar. It's almost always a sugar if it ends in ose. Cellulose, fructose, sucrose, glucose, so on and so forth. Now, this is what I was talking about with this external structure called glycocalyx. It's a sugary coating around the bacterial cell, a bacterial cell. Now, depending upon whether or not that coating is tightly wrapped around the bacteria or loosely wrapped around the bacteria, will determine whether or not it's considered a capsule or a slime layer. So, for instance, this right here, notice this red coating is very tightly attached to these other layers. This would be considered a capsule because it's tightly wrapped around the cell. Here, you can see that this grainy stuff around the cell, all this grainy imagery right here, that's the glycocalyx around a cell under a microscope, and it's not tightly wrapped around the cell, so it's considered a slime layer. What's important about glycocalyx, it's usually sticky, it's outside the cell wall, it has some of those functions that we talked about in the previous slide. Now, pathogens, some pathogens and some non-pathogens will be able to stick to surfaces because of a glycocalyx. A perfect example is what we call biofilms. Biofilms are basically layer upon layer of bacteria that form and they're stuck together because of the glycocalyx. Now, on a daily basis, each of us has a biofilm forming. It's what forms on our teeth throughout the day. So, I encourage you, at some point during the day, take your fingernail and just scrape the front of your front tooth with your fingernail. You'll probably see a white film that comes off of your front tooth. That essentially is a biofilm. It's bacteria, also mixed with a little bit of food particles and stuff, but that is a bunch of layers of bacteria that have built up on your teeth. Now, this is part of the reason why we brush our teeth, is because bacteria form these biofilms, and just rinsing our mouth with water doesn't work. These biofilms are sticky, so they attach to a surface very tightly, and it's difficult to remove them, which is why we have the mechanical removal via brushing our teeth. And then also with flossing is also mechanical, and then mouthwash is chemical, but it loosens and removes. Capsules also, so slime layers allow for this biofilm to form. Capsules help prevent a process called phagocytosis. Our white blood cells scan any cell they come in contact with and look for these special tags. Every cell in our body has a tag on it. And it's just like at the grocery store when you go to Walmart and you go to self-pay. You just scan it across the machine, and then it beeps, and you see the name of what it is. Now, granted, when a white blood cell scans something, it doesn't beep and then show a list of what it is. It scans it for the tag, though. And if the tag is missing, then it treats that cell as foreign, and it will attack, and it will engulf via phagocytosis. Basically, it eats that cell. So our cells all have tags on them, which our immune systems recognize. They go through and they see our cells and they scan them and they say, oh, I know that tag. That tag's fine. Move on. And then until it comes in contact with something that doesn't have the right tag or no tag at all, then these white blood cells will then attack. Basically, it's the white blood cells' way of seeing what's foreign and what's not foreign. Excuse me. All right. Hopefully everyone's clear on this. Capsule is the glycocalyx wrapped tightly around the outside of the cell. Slime layer is loosely around the cell, and it helps create a biofilm. Lipids. Lipids include fats, oils, waxes, and steroids. Sterols associated with lipids are really important as well. They are vital to cell membrane structure. We'll get to that. Lipids are considered organic because they have carbon, hydrogen, and oxygen, but they are hydrophobic. We talked about how many of these fats or lipid structures are hydrophobic or even possibly amphipathic. They have partly hydrophilic and partly hydrophobic regions. Lipids, for the most part, hydrophobic. That's why oil does not mix well in water. It separates. This is why butter doesn't dissolve in water, because they're hydrophobic. This is what a lipid structure looks like, what we call a saturated or unsaturated fatty acid. Now, saturated fatty acids means that every single carbon in the tail is full with 4 bonds. So anyone you look at, here's 1, 2, 3, 4. 1, 2, 3, 4. Every single one of them has 4 bonds. That allows the tail to be straight. And then it attaches to this up here, which is what we call a glycerol molecule. Glycerol is the head portion of a phospholipid, while the fatty acid tail is the tail portion. Now, if it's saturated, the tails are straight. If it's unsaturated, there is a double bond somewhere in the tail structure that forces it to bend in a an odd direction, which means the tails cannot pack tightly together, which is why unsaturated fats are typically liquid, such as olive oil, peanut oil, any type of liquid oil you can think of is most likely unsaturated because of the fact there's a double bond somewhere in the tail of a fatty acid chain. A saturated fatty acid, since the tails are really straight, they can stack together really neat and tightly, which allows for them to be solid at room temperature. Butter is a perfect example of that. All right. Some functions, energy sources, cell structure components, and signal. We'll talk mostly when it comes to lipids about cell structure components, how it's important for the cell membrane and then other pieces in the cell membrane, right? The phospholipid bilayer. These are just some examples. Phospholipids, those have the glycerol head and the fatty acid tail. They make up the cell membranes. Now, glycolipids and lipoproteins, they're simply lipids attached to something else. So glyco is going to be short for carbohydrates. So glycolipids are lipids or oils attached to a sugar. Lipoproteins are lipids or oils attached to a protein. Okay? These can act as tags on the outside of the cell. So sometimes when the white blood cell comes along, it's looking for a glycolipid or a lipoprotein. It scans the cell to find it, these recognition tags. And if they don't find it, they treat it as foreign and they destroy it. A little bit more LPS, lipopolysaccharide. So LPS stands for lipopolysaccharide, meaning you have a lipid, okay, and you have many sugars. Polysaccharide means, poly means many, saccharide means sugar. Lipo is lipid, so lipid plus many sugars. This is going to be found on the outside of gram-negative bacterial cell walls. We call it the LPS layer. Gram-positive cells don't have this. Gram-negative do. Now, I know we haven't gone over gram-positive versus gram-negative yet. That's okay. We will I don't think it's this chapter. It might be the next chapter. We'll go into a lot of detail with gram reactions. Gram-positive versus gram-negative. Because in lab, you're going to have to know the difference. Mycolic acid is unique. It is unique to specific organisms. For instance, tuberculosis, Mycobacterium tuberculosis. The outside, instead of having an LPS or peptidoglycan, instead it has mycolic acid. Now, mycolic acid makes tuberculosis extremely pathogenic. It's one of the pathogenic traits that it has. And when we talk about tuberculosis later in the semester, we will talk specifically about the mycolic acid as part of the cell wall. And then cholesterol. Cholesterol is also found in cell membranes, especially animal cells. Here's a really good example of what a phospholipid bilayer looks like, or the cell membrane. Now, the cell membrane is essentially the same thing as a plasma membrane. The terms are interchangeable. So, a bacterial cell has a cell membrane or plasma membrane, followed by a cell wall, most of them, not all of them, and then sometimes followed by either a capsule or a slime layer. Whereas animal cells, all our cells have is a phospholipid bilayer cell membrane, or plasma membrane, whatever you want to call it. Either one's acceptable. So the big difference is our cells, animal cells, have only a phospholipid bilayer holding our entire cell together. Every cell in our body is held together by two molecules thick, a 2-molecule thick structure. Here's molecule 1 facing inward, molecule 2 facing outward. That holds our entire body full of cells together. Every cell is held together this way. It's just, to me, one of the most interesting things in all of biology, that humans rely on 2-molecule thick structure to allow our cells to survive. It's very interesting. I digress. So you can see here that the bilayer has some things in it. It has some proteins embedded in it. There will be some other components that will stick off of the cell wall, like recognition tags, along with adhesion structures, allowing them to stick to other things. But the point here is just to show you that the cell membrane, or plasma membrane, has not just phospholipids, it also has proteins embedded. Also, it'll have some lipids embedded as well. In the middle, where the tails are facing each other, you'll actually find some sterols, like cholesterol. You'll actually find that in the middle here. It's a non-polar molecule. It's hydrophobic, so it can't go on the outside because there's water. Can't go on the inside because there's water. So it'll go right in the middle where there's no water. Okay. All right. Moving on to nucleic acids. So nucleic acids include DNA and RNA. These are the two we're going to focus on. DNA and RNA serve as basically the genetic material for cells and viruses as well, which are not considered living, but they do have DNA or RNA. There's two major categories of nucleic acids. I've already kind of said it twice already. DNA versus RNA. Now, DNA is a double-stranded helical molecule. Basically, it's a winding staircase in structure, its structural form, where RNA is a single-stranded molecule. Now, these are not the only two differences. There are other differences as well, but every cell that we have has almost every cell that we have has DNA in it. It's our genetic code. It codes for everything. DNA codes for everything. Every protein that gets made in your body is encoded for within the DNA in your nucleus. Now, bacteria don't have a nucleus because they're prokaryotic, but they still have DNA. DNA just happens to be free-floating in the cell, and it kind of coils on itself and twists and turns to take up less space. RNA is kind of an intermediate storage molecule, because you'll go from DNA to RNA to a protein. That's what we call the central dogma of biology. It's the genetic flow of information. DNA, RNA, protein. So the way I like to think about it, and the way I've thought about it for 15, 16 years, is DNA is a cookbook which has all the recipes that you have and know of. Okay, all of them. And then you decide, all right, today I'm going to make a random recipe. And you pull one out, and it's, I don't know, brownies. So the DNA, right, is the cookbook with all the recipes. When you pull out a specific recipe, that's a gene. And when you start to read the genes, you use amino acids to build an RNA copy of the recipe. So basically, you are taking the recipe and putting all the ingredients together in a bowl, and that's your RNA. Okay, it's your shorthand version of the original sequence or the original recipe. Then you bake the brownies. Your final product is a protein. The brownies are your functional proteins. So when it comes to this, what I really need you to know, DNA goes from DNA to RNA to proteins. That is called the central dogma in biology. And this is not just in humans. This is also in bacteria as well. So the monomer for nucleotides or nucleic acids is nucleotides. And they have a really unique structure. And I'm going to skip this slide because I think I have an image on the next one. Yeah. I'd rather show you this and talk about this one. All right. So when we look at this, the piece up here at the top is the general structure of a nucleotide. A phosphate, sugar, and then some nitrogenous base. This is going to stay the same for every single nitrogenous base, with the exception of this nucleotide, or the base pair. So you'll still have a phosphate, you'll still have a sugar, but the actual nitrogenous base will change. Now, it depends on whether you have DNA or RNA. If you're looking at DNA, then it has a specific set of nucleotides. If you're looking at RNA, it has a specific set of nucleotides. And they vary between the two. So right here in this middle image, you have RNA on the left side and DNA on the right side. How do I know this? Well, besides the fact that the text is written here, let's pretend that's not there, I can tell that this is RNA versus DNA just by looking at one specific spot. And that's what we call the 2' carbon. We count carbons clockwise. So here's 1, 2, 3, 4. 1, 2, 3, 4. And I can tell just by looking at whether, just by looking at the 2' or the second carbon, whether it is DNA or RNA. How do I know that? Well, if we look at RNA, notice on the 2' carbon there is an OH group. But if we look at DNA on the 2' carbon, the oxygen is missing and all you have is a hydrogen. Sorry. This is why DNA is named in this fashion. Deoxyribose nucleic acid. Deoxy means without oxygen. It's missing an oxygen. So the name tells you that it's missing an oxygen. And it's missing it on the 2' carbon, the second carbon. 1, 2. Instead of having an OH group, it only has a hydrogen. So we know this is DNA. Looking at this one, it has an OH group. That tells me it is RNA because it's not missing the oxygen. Now, the third image here, or box, is the 5 different nitrogenous bases. As I said, phosphates, sugars, and nucleotides, the phosphate and the sugar, stay the same. The nitrogenous base changes. For instance, you can have cytosine, thymine, guanine, adenine, or uracil. Now, each one pairs specifically, and 3 of them are shared between RNA and DNA. RNA and DNA both have cytosine, guanine, and adenine. Both DNA and RNA have both cytosine, guanine, and adenine. The 4th nitrogenous base will change depending upon whether you're looking at DNA or RNA. If you are looking at DNA, the 4th nucleotide is thymine. Which is right here. If you were looking at RNA, thymine is replaced by uracil, which is this central one right here. This is uracil, which is found in RNA. Now the pairing is still the same. So cytosine and guanine pair, they match, and then in DNA, adenine and thymine match. Or if you're looking at RNA, you have adenine and uracil matching. Cytosine and guanine will always match, but if you're looking at RNA, you have adenine and uracil. If you are looking at DNA, you have adenine and thymine. Okay, so thymine and uracil are interchangeable depending upon whether you're looking at RNA or DNA. So I have just gone over the monomer of a nucleic acid, a nucleotide, but I've also shown you key differences between RNA and DNA, between genetic material. I do expect you to know this. Okay? I could ask, there's probably about 10 different questions I could ask about this stuff. So pay attention to it. Make sure you took good notes. If you have to listen to it again to know the differences and how I tell what's different about each of them. This is just reiterating what I was saying. This one contains, DNA contains adenine, guanine, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil. So again, the last one in this list is interchangeable depending upon whether you're looking at DNA or RNA. So if you're looking at DNA, it's thymine. For the 4th one, for RNA, it's uracil. Now, one of the things that I mentioned with DNA is it's helical, meaning it's spiral, like a spiral staircase, and it has 2 rungs to it. So it has one side and another side, and they face each other, and the nucleotides pair in the middle. So you have one side, the other side, and then guanine facing this way, and then the other strand will have cytosine facing that guanine. And what will happen is, to hold the strands together, bonds will form between the pairs. But not just any bonds, some very specific bonds. You will have hydrogen bonds form between the pair, the nucleotide pairs. So if you have a guanine and cytosine, then you have 3 hydrogen bonds holding it together. If you have adenine and thymine, you have 2 hydrogen bonds holding it together. Now, RNA is single-stranded in most cases. Almost always. There's some exceptions. So we don't have to worry about bonding with that. But with DNA, since you have 2 strands that need to be held together, you have to have some type of bond holding those strands together, and they're hydrogen bonds. Adenine and thymine have 2 of them. Cytosine and guanine have 3 of them that hold them together. This is just showing you how DNA and RNA are built. We create a backbone, a phosphate backbone. And you can see we're using dehydration synthesis, right? Losing a water to build something and create bonds. So we create a phosphate backbone. Here's the base facing inward. You would have another strand facing these bases right here. Okay. So overall, as long as you know the difference between DNA and RNA, you know that hydrogen bonding holds the strands together in DNA. There's 3 hydrogen bonds for cytosine and guanine, 2 for adenine and thymine. Should be all right. Now, a lot of times I get people asking me to give examples of questions I would ask. That's really what the participation assignment is for. So make sure that you're, after you watch this, you attempt the participation assignment. You only get 1 attempt, but if you took good notes, you should be fine. And any questions you get wrong, I recommend you go back and look at that specific spot in the video to try to understand that topic in more detail. Again, use the book as well. That's why you have it. I will give you a little example. If I'm looking at a piece of DNA and it has 50 pairs, so 50 paired bases, and of the 50, 13 of them are adenine and thymine, okay, and 37 of them are cytosine and guanine. Now, if we think about that for a second, that strand has lots of extra hydrogen bonds, because remember, cytosine and guanine have 3 compared to adenine and thymine, which only have 2 hydrogen bonds holding it together. So if you were to compare that to another strand and say, all right, well, I have another strand of DNA and it's the exact opposite. I have 37 adenine thymine pairs and only 13 cytosine and guanine. Well, since there's fewer cytosine and guanine pairs, the first strand that we talked about would be much more difficult to separate, because we do have to separate them. Bacteria, cells, we do. We have to separate DNA at certain points. So the more cytosine and guanine pairs you have, the more difficult it is to separate the strands. All right, so that's that's the type of question I would have no problem asking on an exam or a quiz. It tests your understanding. And as I mentioned on the first day of class, that we're going to be testing concepts. We're not going to just memorize and regurgitate information. Yes, there is a time and place for that, and there will be some of that in this course. For the most part, though, it's going to be application-based. I am going to ask you to apply concepts, which will be very important. And you may hate me for it, but I promise you, when you get to whatever program you're going to move on to, they will do the same, especially if you're nursing. It's concept-based. The NCLEX is case study-based. So knowing how to answer questions that are application-based is very important. And we'll get lots of practice with that this semester. All right, I'm going to move on. This, just some functions of nucleic acids. We know that DNA, as I said, is the cookbook. It has the genetic blueprint of all cells. RNA directs the production of proteins. There are specialized RNAs. Don't worry about them, though. And then ATP, for instance, is an energy source, but it's considered a ribonucleotide. Don't worry about specialized RNAs in terms of ribozymes or double-stranded RNA. Don't worry about that. Your book may mention that. Don't worry about it. We will talk about transfer RNA and messenger RNA later. but not in a ton of detail. All right. Proteins. Proteins are basically everything to us. They are the end product in the central dogma, and it carries out a function, whatever that function is. So at every level, a protein is involved in some fashion, whether that is building a cell membrane or moving molecules in and out of a cell, or activating other molecules. Proteins have a very diverse range of functions, especially in humans. We have them for structural components. We have them for important attachment mechanisms. Proteins are really, really, really important. The monomer for a protein is an amino acid. There's 22 of them that we need. Humans, we need 22 of them. We can't make all of them. So let me take a step back for a second and clarify something. When I say 22 amino acids, that includes all of them. We need 20. So there are technically 22, but we're not going to worry about technicalities. We need 20 of them. Okay, we can't make them all though. We need help from bacteria and other and food to give us those essential amino acids. So I want to make sure it's clear that technically, yes, there are 22 of them that are known, but for humans, 20 is what we want to focus on. 20, but we can't make them all. We can only make a fraction of them. So we're required in our diet as well as bacteria in our digestive system to help make some. The protein structure of an amino acid, right, the monomer, has this general structure. An amino group on the left side, a central carbon, and then a carboxyl group on the right side. Also, you'll have a hydrogen attached to the carbon, and then an R group attached to the other side of the carbon. R group simply stands for the remaining or side groups or the rest of the amino acid. It could be as simple as another hydrogen, or it can be really complex and have lots of ring structures. Like tryptophan, for instance, has like 3 or 4 rings. It's important to know that the general structure, though, always remains the same. Amino group, central carbon with a hydrogen attached to it, and a carboxyl group. The R group, the side groups will change, but the rest of that will stay the same. Okay, so that amino acid structure, it follows for the most part, the nucleotide structure, right? Most of it stays the same with only one component changing, right? So just like nucleotides, most of the structure stays the same, and a single region changes. So amino acids, most of it stays the same: amino group, central carbon, carboxyl group, and the hydrogen off of it. But the R group changes. Now, I will not expect you to memorize the amino acids. There's a whole table full of them. I won't expect you to memorize them. If we talk about anything associated with amino acids, I will give you the name of the amino acid. Now, proteins are made up of just chains of amino acids. That is considered the primary structure. So let's say there's 50 amino acids in a row, they're all connected. That's your primary structure of a protein. Then you end up with a secondary structure where the protein will then take on 1 or 2 forms. A helical form, a winding staircase form, or a what we call a pleated sheet. So you have an alpha helix or a beta pleated sheet, where you have these individual kind of tails almost that line up. And what holds them together is hydrogen bonds. So just like DNA, proteins in their secondary structure are held together by hydrogen bonds. Hydrogen bonds are really, really, really important. They're used to hold DNA together, hold proteins together. They're used to link water molecules together. So having hydrogen bonds is very important. It helps, in this case, retain the structure. The alpha helix will stay coiled. The beta sheets will stay pleated like this because the hydrogen bonds will hold them in that shape. Then you get to the third level where you start to have interactions between your alpha helices and beta sheets. And then you get to your quaternary, which is the last level, the most complex level. And this is where all the different pieces have come together, and then the protein has folded into a functional shape. Proteins must fold into a very specific shape to function. Let me repeat that. Proteins must fold into a very specific shape to function. Now, depending on the protein, that shape is different. But if they don't fold into that shape, it's not going to function. And that is definitely not a good thing. If you have a non-functional protein, then that could be pretty serious. For instance, if you were to think about a disease called cystic fibrosis in humans, simply it's a protein channel that's damaged that causes cystic fibrosis. So if the protein was folded properly and not damaged, cystic fibrosis wouldn't happen. But in that case, the protein is damaged. Now, one of the things with proteins as well as DNA, temperature can affect them. So hydrogen bonds are pretty strong. They're actually pretty strong. But if you increase the temperature too much, if you heat a protein for too long, the hydrogen bonds break and the protein unwinds. The hydrogen bonds break and the protein unwinds. At that point, it doesn't have the proper shape and therefore isn't functional anymore. It's useless. So it's important to know that temperature has an effect on protein structure. If it's too high, it'll break the hydrogen bonds. Same thing with pH. If pH is too high, it'll also break the hydrogen bonds. Lots of things can affect protein structure, and the worst thing that could happen would be the hydrogen bonds breaking, unwinding that protein, making it non-functional. This is just a small snip of how many functions a protein has. It's very, very, very tiny. Enzymes, which we'll talk about in a minute, scaffolding in cells, what we call structural scaffolding, cellular transporters, they allow things to move in and out of the cell. Cell recognition, I talked about the tags, and communication. They can create communication molecules. These are just some of the functions. There are a lot more than this. All right. Here's an example of a structure that is protein-based. Basically, it's made up of a series of proteins. So flagella. Flagella are these long filaments that help bacteria move from one area to another. They propel themselves. Not all bacteria have them. Some do, but others don't. Humans, the only cells that have true flagella are sperm. Everything else typically has either cilia or microvilli, something similar. But flagella, they're long, they're made up of flagellin proteins, and they kind of spin. They're outside the cell wall, but you can see how they're anchored in multiple places in the plasma membrane, in the peptidoglycan, so in the cell wall, and then in the outer membrane as well. So this is a gram-negative cell, meaning it has a cell membrane, a cell wall made of peptidoglycan, and then another outer membrane, the LPS layer, the lipopolysaccharide layer that I mentioned. So you can see that this is embedded in every single one of those layers. And that's important because the filament moves very fast and kind of in this spinning motion, which is kind of violent on the cell, right? It's going to spin very quickly and it's going to pull and stretch, and it has to be anchored very strongly to the cell, otherwise it will break off. As I mentioned, they're made up of flagellin proteins, and it's just a bunch of them together, a bunch of flagellin proteins together. Flagella are pretty interesting. There are many different orientations of flagella, which we'll talk about in this class. Doesn't matter, they can rotate clockwise or counterclockwise, but it propels them forward or in a given direction. So this is what I was talking about with arrangements. You have what we call monotrichous, one flagella on one end of the cell. You can have amphitrichous, where you have flagella on both ends. Lophotrichous, this is what we call a tuft of flagella, and you could have it on one or both ends. So think of a mop, essentially. All of the individual strands of the mop, they meet at a given point on the handle, right? That would be considered what kind of a similarity between lophotrichous tufts would be looking at a mop where the individual strands, they all come together to a given point on the handle, right? So it looks kind of like a tuft, or you could say a broom. All those fibers meet at one given point, and then the rest of it spreads out, fans out. It could, in lophotrichous arrangements, it could be a tuft could be on one end or both ends. It really depends on the organism. And then peritrichous. Peritrichous means flagella on around the cell completely, all over the cell. There's no particular order to it. It's just on the cell in pretty much any arrangement and in any position. So monotrichous, one flagella on one side. Amphitrichous, a flagella on both sides. Lophotrichous, a tuft on either side or both sides. You could have one or both. And then peritrichous, where you have flagella covering the entire organism. All right. Flagella are used for motility. They use the run and tumble method, which is really funny. Basically, they spin the flagella really fast, propel themselves forward, and they don't really have a mechanism to stop. So the only thing they can do is stop spinning the flagella. But their momentum will carry them forward, and they'll flip over end over end over end until they come to a stop, essentially. And then they'll spin the flagella again, and then they'll stop spinning them, and then momentum will carry them forward, and they'll flip over themselves again. So it's called the run and tumble method. It's actually really funny. So what would be the advantage of motility, right? If you're a bacteria, what's the advantage of motility? Well, let's say you live in an area where there's very little nutrition, right? Very little nutrients. So you can move to a different place, right? You can move toward or away stimuli. Some bacteria move toward or away from sunlight, or certain chemicals, or even magnetic fields. They can move toward or away magnetic fields. If we were to ask chemo versus phototaxis, so taxis is movement, so chemo is movement towards a chemical or away from a chemical, and phototaxis would be light, so movement to or away from light. Oops. So, advantage of motility. They can move, which allows them to find better places to live if their environment's not favorable. They can move toward or away from stimuli. And then two examples of that are chemo and phototaxis. Whether if it's chemotaxis, they're moving towards or away from a chemical, or if it's phototaxis, they're moving toward or away from light. And they use the run and tumble method. I will try to find a video of it and share it with the class, but I haven't been able to find a really good one in a while. Another way to move, axial filaments. So bacteria that are spiral shaped, what we call spirochetes, they have many of them have axial filaments. Basically, there's a filament, and it's hard to see. You can see two red lines right there that go down the middle of the cell, and then this endoflagella, the yellow, is coiled around it, and it kind of acts like a spring and a corkscrew motion. So it's anchored at one end, and then it'll allow for the endoflagella to spin around this central filament here. So this central filament here, I know it's probably really hard to see, but there are two red lines in the middle of these, the yellow, and that's your central axial filament, or your axial filament. And basically one end of the yellow endoflagella is anchored, and then it spins around the axial filament. It moves typically in a corkscrew fashion, which for some pathogens, that's really important because it allows them to burrow into our cells. For example, syphilis. Syphilis is caused by a spirochete, and it can burrow into brain tissue if it's untreated for years. Or it can move into different parts of your body and kind of burrow its way into cells. There are other pathogens that can do this as well that are not spiral-shaped or spirochetes, but these ones have a really distinct advantage in that they can use that corkscrew motion to get themselves into a cell or between cells. So now that we've talked about the axial filaments and the flagella, which are types of, they're structurally made of proteins, we're going to talk about enzymes, which are also proteins. Okay, so as I mentioned, enzymes are really important, and we were going to talk about them later in the chapter. So we finally come to that. It's important to understand that these structures, okay, they're proteins. They're based on a protein structure. So we'll talk about each piece, and then we'll talk about the whole thing. The first thing we need to realize is that the individual pieces have very specific names, and they are they function or don't function based on their structure and whether or not they're interacting with other things. So first off, here, this first image, this is the apoenzyme. It is the protein portion of the enzyme that is inactive. So in this form, it's not active. It's not working. But as soon as you add a cofactor, which is not protein-based, you add that coenzyme or cofactor to fit into the active site of the enzyme, it now becomes what we call a holoenzyme. Holoenzyme. The whole enzyme, meaning it has both the protein portion and the non-protein cofactor. And when it has both of those, the enzyme becomes active. And what it does is it will either help speed up or slow down chemical reactions. If we want to speed up chemical reactions, we can use enzymes. If we want to slow chemical reactions down, we can also use enzymes, or we can block enzymes from being used. So this is just showing you how mechanistically the enzymes work. You have your active site, which is where the substrate binds. The substrate is anything that the enzyme works on. And enzymes are pretty specific. They'll work on a pretty specific given substrate. They're not really like one size fits all and works on anything. They're a bit specific. But the substrate binds to the active site, and then in this case, it breaks that substrate down, and here are the products. It also works the other way around. If you add these two pieces to the active site, it can put them back together. So it works both ways. It can either break things down, it could create things, it can put them together. So it kind of works both ways. Okay. So this is the mechanism behind enzymatic action. Now, this shows you that with enzymes, just like I mentioned in proteins in general, temperature affects enzymatic activity. So now, this image shows just generally how this protein has folded properly, it's in its functional shape, it's working perfectly fine. All of a sudden, something changes, temperature too high, pH too high. The enzyme can be denatured, which means it'll be broken down. Okay? The enzyme will be broken down, or the protein in general. But all these things here, temperature, pH, substrate concentration, inhibitors, all have an effect on the enzymatic activity. How fast the enzyme works, or if it works at all. If the temperature gets too hot, the enzyme will denature. It'll break apart and it becomes non-functional. If the pH strays too far in one direction or the other, it will break the bonds holding the enzyme structure together, the protein structure. Hopefully you know which bonds those are. Right, what bonds hold proteins together? Hopefully you answered hydrogen bonds. Okay? Hydrogen bonds hold them together. But if we stray too far, then we denature proteins. We break them down and they unwind and then they're useless. This is an amazing image, figure 5.5a. Actually, I think this is from a different text. I apologize. I will post this image on Blackboard because this comes from a different text. I know your book has an image that's similar to this, but I will post this slide. You can see here y-axis, enzymatic activity, x, temperature. Every enzyme has an optimal temperature or optimal range that it works at, that it works most efficiently at. Humans are enzymes. work really efficient at 98.6 degrees body temperature, right around there. And granted, there's a little bit of there's wiggle room. It doesn't have to be perfect, but you can see, there's a slight range in which the optimal temperature works. Anything below that, the efficiency drops. Anything above that, it will denature the protein and destroy it, and it'll make it useless. So there's always a optimal temperature that enzymes work at, an optimal pH that enzymes work at, an optimal salinity, basically salt concentration, that enzymes work at. So there's very specific criteria that make enzymes work at an optimal functionality. But if you change one of those, then it can drastically affect how efficient that enzyme is. And this shows a really good example of that. Lower temperature, lower efficiency. As you move up the range here, there's the optimal range. If you stray too far above it, it denatures and the activity of the enzyme goes down to 0 because it denatures. It unwinds. It's useless after that. Now, when it comes to speeding up or slowing down reactions, we've talked about speeding them up, right? Adding an enzyme to speed it up. Now, I want to talk specifically how an enzyme will slow down a reaction as well. But before I start speaking about how we slow the reaction down, I want to talk about the specific mechanism that speeds the reaction up. So hopefully you recall earlier I mentioned something called activation energy, right? It's the X amount of energy required to start a reaction. I used an example of a campfire with matches and then matches with gasoline. The activation energy, again, is the amount of energy needed to start a reaction. Every reaction has this. To speed up a reaction, one of the ways we can do that is adding an enzyme. When we add the enzyme, what it does specifically to speed up the reaction is it lowers the activation energy. So the amount of energy it needs to start that reaction drops. Now, it doesn't go down to 0. It's still going to require energy, but it's going to be less than without an enzyme. So let's say a reaction requires 100 units of energy to start. That's its activation energy without an enzyme. You add enzymes to that system, and then all of a sudden, the enzymes can speed that reaction rate up by lowering the activation energy. So instead of 100 units of energy, now maybe it needs half that, 50. So by lowering the activation energy, in response to that, the reaction speeds up, right? The reaction can happen faster. So if it took 100 units of energy to start, right, it would go 100 units, and then it would start. 100 units, and it would start. But now it's 50, 50, 50, 50, and it goes faster and faster and faster. Okay? This is really good when the cells need it. If they're low on glucose and need glucose immediately, they can speed up the production of glucose. But, as I mentioned earlier, we don't always want enzymes to be working at full speed all the time. Sometimes there's too much of a product and we want to slow the reaction rate back down. We want to slow it back down. And there's several ways to do this, but I'm going to talk about two really common ways. The first one is competitive inhibition. Essentially what happens is an inhibitor, usually the product itself, will bind to the active site of the enzymes, and it will block the substrate from binding. So remember, The substrate binds to the active site, the enzyme does its job, perfect. Everything's working great. But now we have too much of that substrate. There's too much of that product. We don't want to make more. Or we want to slow it down, the amount that's produced. Well, the inhibitor will actually bind to the active site and physically block the substrate from binding. And if the substrate can't bind to the enzyme, the rate of the reaction will start to go down. It'll slow down because the enzymes are no longer working on the substrates because the substrate can't bind to the active site. So by doing this, competitive inhibition, meaning they compete for the active site, the substrate and the inhibitor, and if the inhibitor binds, it will literally block the substrate from attaching, which will then slow down the rate of the reaction. Then you have non-competitive inhibition. Non-competitive inhibition is a little bit different. It binds to so this is normal binding on the left. On the right, what happens is the inhibitor will bind to a site other than the active site. It'll bind somewhere else. The site is called the allosteric site. And you can see on the left, it's this little area that's not filled in because this is regular binding. But now they want to slow the reaction down, so the inhibitor binds to the allosteric site. In response to binding to the allosteric site, the active site changes. The shape of the active site changes. And if you recall, I said earlier that enzymes will work on a couple different substrates. It's not one size fits every single substrate. So the active site is pretty specific. So what happens is the altered active site, which has been changed by the fact that the non-competitive inhibitor binds to the allosteric site, this substrate just bounces. It'll hit the enzyme and just bounces off of it because it can't fit into this new active site. It's not the right shape. So this will bounce off of the enzyme. And therefore, if the substrate can't be worked on, it'll then slow the reaction back down. Now, both competitive and non-competitive inhibition are reversible. So the inhibitors can let go, and then the enzyme can speed the reaction back up. So this isn't a permanent thing. This is, right now, we need to slow the reaction down because we have too much of a product. But once they use up that product, and there's very little left, the inhibitors will let go, and then all of a sudden the enzymes can do their job and speed up the reaction again. Enzymes are pretty incredible in the way they function. And we'll talk more about enzymes later in the semester, especially when we talk about diseases caused by certain bacteria. And this image is interesting. It's called the negative feedback loop. Again, this is from a different text as well, I believe. So I'll post this one with the other slide. You have your substrate. This is the normal pathway, right? It binds, it's worked on, there's your product. That's a normal pathway. But if there's too much of this product, notice there's way too much of the product over here, and they want to slow down the reaction, the product itself will act as the inhibitor. In this case, non-competitive inhibitor. But it changed the active site and the substrate can't bind, which will slow down the reaction. Okay, so this is considered what we call negative feedback loop or negative feedback inhibition. Basically, there's a change that will slow the reaction rate down. Excuse me. So this is a great example of how non-competitive inhibition works. Shows you the normal pathway on the left, then all of a sudden you get too much of the product, and the product acts as its own inhibitor to prevent more of itself being produced. But once all that extra product gets used, it will let go of the allosteric site, and the active site will change back to its normal, original structure. And then the enzyme can do its job. And it will work on the substrate and speed them up. Okay, so that's quite a bit about enzymes, but enzymes are super important later in the semester when we talk about diseases of the organ systems, because certain bacteria can use enzymes to harm our cells. And in response to that, our cells use enzymes to try to harm the pathogens. So it's really important that we know that enzymes are important, how they function, how they play a role in gene expression, meaning speeding up or slowing down the rates of reaction. All of that is pretty important to understand going forward. As you can see, we've finished this chapter, chapter 2. Please make sure you take notes. You start, once you finish the lecture, take notes, do the participation assignment for chapter 2. This way you can test yourself on the material, and that will help you identify areas that you may need to focus on. I have office hours this week, so make sure if you have questions to come to them. Times and dates are on the syllabus. All right, everybody, have a great day, and I will talk to you all soon.